Have you ever taken a second to wonder what is happening inside of our bodies throughout the day? Let's take a gander at Noe here. Here he is during his daily run at the park. What is happening here? How is he able to move like that? Let's take a closer look.
Working together are his skeletal, muscular, nervous, cardiovascular, and respiratory system. Today we're going to be discovering parts of his skeletal, muscular, and respiratory system. First, let's identify the major bones he is moving here. Inferiorly, the bones Noe is using here are his femur, tibia, fibula, tarsals, metatarsals, and phalanges, as well as his pelvic girdle. The pelvic girdle is composed of two hip bones that articulate with the sacrum and coccyx to form the pelvis. The pelvic girdle supports the trunk of his body, protects his interior organs, and provides the range of motion you see here through a ball and socket joint connection with his femurs. Superiorly, Noe is controlling his humerus, radius, ulna, and the carpals, metacarpals, and phalanges of his hands, as well as his pectoral girdle. Like the pelvic girdle, his pectoral girdle allows for a range of motion to occur in multi-axial fashion, so his arms are able to swing by his sides, helping to conserve energy. Let's not forget about his spine. Noe's spine contains masses of fibrocartilage called intervertebral discs. These discs act as shock absorbers, making his running experience comfortable, not to mention safe. Now let's focus on his muscles.
Noe is moving his skeletal muscles he controls consciously. What is the anatomy of his muscles? Muscles are organs composed of specialized cells or fibers that use chemical energy to pull on the bones they are attached to. Each muscle contains bundles of fascicles, and within each fascicle are numerous muscle fibers or muscle cells. The working components of a muscle fiber are the myofibrils and the sliding action of the thin and thick myofilaments that make up the myofibrils. Thick filaments are also known as myosin filaments. Myosin filaments are composed of two twisted protein strands with projecting globular parts called heads. Thin filaments are also known as actin filaments. Actin filaments contain a helix of actin molecules as well as the proteins troponin and tropomyosin. Troponin is a binding site for the myosin heads, while tropomyosin covers this binding site. In order for a muscle contraction to occur, a neurotransmitter must stimulate it. Acetylcholine is released into a neuromuscular junction. Synaptic vesicles form the end of a motor neuron, are transported across the synaptic cleft to the motor end plate of the muscle fiber. Once acetylcholine is bound to specific receptors, the muscle fiber's membrane or sarcolemma becomes permeable to sodium ions. The entry of charged sodium ions passes an electrical impulse through the transverse tubules of the fiber all the way to the sarcoplasmic reticulum. The sarcoplasmic reticulum responds to the influx by diffusing calcium ions into the sarcoplasm of the muscle fiber. The released calcium binds to troponin, simultaneously pulling the tropomyosin aside and exposing the binding sites of the actin filament. Exposed binding sites on actin allow the muscle contraction cycle to occur. The myosin head bind to actin, forming cross bridges connecting myosin to actin. Upon binding to actin, myosin heads spring from their cock position and pull on their actin filaments in a power stroke fashion, releasing ADP and a phosphate in the process. A new ATP binds to myosin, breaking the connection to actin. The ATP breakdown provides energy to cock the unattached myosin heads and store energy for the next power stroke. As long as ATP and calcium ions are present, the cycle continues. When calcium concentration is low, the muscles become relaxed. The sliding filament model is what allows Noe to run across the field. Keep up the pace, Noe!
Now, what is happening to his oxygen levels as he runs? Let's explore. First, we must examine what our lungs are up to during rest. During normal respiration, Noe would breathe in about 500 millimeters of air. Normal resting inhalation means the muscle fibers in the diaphragm below the lungs contract, moving the diaphragm downward, enlarging the thoracic cavity and lowering the intra-alveolar pressure, causing air to rush in. The external intercostal muscles between the ribs may also contract then. Resting exhalation means those tissues return to their original shape due to the elastic recoil and surface tension, causing the thoracic cavity to decrease in size while increasing intra-alveolar pressure, essentially pushing air out. However, during exercise, additional air is being inhaled and exhaled, so extra muscles are recruited. During forced inhalation, the diaphragm and external intercostal muscles contract more forcefully. The sternocleidomastoid and the scalene muscles also pull the thoracic cage further upward and outward, enlarging the thoracic cavity and further decreasing the intra-alveolar pressure. Conversely, during forced expiration, the internal intercostal muscles can contract, pulling the ribs and sternum downward and inward, increasing the air pressure in the lungs to force more air out. Why does Noé breathe harder the longer he runs? An increase in carbon dioxide production increases carbon dioxide elimination. As Noé exerts himself, his cells are increasing their production of the byproduct carbon dioxide. Carbon dioxide in turn reacts with water to produce carbonic acid. As the carbonic acid dissociates, the concentration of hydrogen ions increases, and the pH of the internal environment drops. This increase of hydrogen ion concentration in the cerebrospinal fluid stimulates chemoreceptors in the respiratory center. The respiratory centers are three pairs of loosely organized nuclei in the reticular formation of the pons and the medulla oblongata. The respiratory center in the brainstem helps regulate the hydrogen ion concentrations in the body fluids by controlling the rate and depth of breathing. The respiratory center increases his breathing's rate and depth, causing his lungs to excrete more carbon dioxide, thereby maintaining a safe hydrogen ion concentration within his body fluids. And that is why Noé breathes harder as he runs harder.
Noé, are you okay?
Oh no, he needs some milk.
No. No. He wasn't ready. He wasn't ready.
Noé fell down. He seems to have cut himself. His tissues are damaged. What happens now? He will need stitches. But let's say that he cuts a small blood vessel. This stimulates vasospasm, the contraction of smooth muscle in the blood vessel's walls. Blood loss loosens almost immediately, and the ends of the severed vessel may completely close. The platelet plug has soon formed, and the blood is coagulating. These platelets adhere to the collagen and to each other and control bleeding for small cuts. Noé looks like he will need a more serious blood clot to halt his bleeding. Noé's damaged tissue have released tissue thromboplastin, initiating a series of reactions that result in a production of prothrombin activator. Prothrombin activator converts prothrombin into thrombin, which requires calcium ions. Thrombin then catalyzes a reaction that joins fragments of fibrinogen into long threads of fibrin, also with the help of calcium. Once fibrin threads form, they stick to the exposed surfaces of damaged blood vessels, creating a meshwork that entraps blood cells and platelets. The resulting mass is a blood clot, which may block a vascular break and prevent further blood loss. The clot also promotes additional clotting as prothrombin acts on clotting factors to form more and more thrombin, creating positive feedback system. Now Noé can heal.